Welcome to AP Biology. Hi, I'm Gail Pfizer from Darnell Cookman Middle High School and Florida State College in Jacksonville, Florida. A lesson overview for today. Today I'll be reviewing Unit 5, Heredity. In fact, it'll be the last two sections of the unit, topics 5.5 and 5.6, environmental effects on phenotype and chromosomal inheritance. By the end of this lesson, you will be able to explain how the same genotype can result in multiple phenotypes under different environmental conditions, explain how chromosomal inheritance generates genetic variation in sexual reproduction, and predict the causes or effects of a change in or disruption to one or more components in a biological system based on a visual representation of a biological concept, process, or model. And you won't need a calculator today, just pen and paper. All right, now for our warm-up, which was yesterday's homework from our, for Friday, it was Friday, Non-Mendelian Inheritance. And I went ahead and filled out some things to save a little bit of time. We have our green is going to be dominant, okay? And purple is recessive with little g, D dominant for dwarf, and D recessive for tall. And I know you've seen this question probably 3 or 4 times already, but there's so many parts to it that we need to cover. It covers everything. All right, so here's chromosome 1 and chromosome 2, and the original questions had a G on one and D on the other of the chromosomes, okay? And a certain species of plant, the diploid number of chromosomes is 4, 2n equals 4. Flower colored is controlled by a single gene in which the green allele G is dominant to the purple allele little g. Plant height is controlled by a different gene in which the dwarf D is dominant to the tall allele D. Individuals of the parental P generation with genotypes uppercase G, G, D, D, and lowercase g, G, D, D were crossed to produce an F1 progeny. I'm going to go ahead and get those back off of there so that doesn't confuse you in any way. Okay, so we have our parental groups. We have big G, big G, big D, big D, and little g, little g, little d is your parental groups. And then so their offspring would be this dihybrid, big G, big D, little d. Now it's asking us to, so that was our cross here. Now our question is, if the two genes were genetically linked, so now we need to not have them on the same chromosome. On different chromosomes, we need to have them on the same chromosome. Describe how the proportions of phenotypes of the resulting offspring would most likely differ from those of the test cross between an F1 individual and a little g, little g. So let's set up our test cross like we did the other day. And so our options were, here was our F1 cross. So we'll have big G, D. We'll have big G, little d. We'll have little g, D. And little g, little d. I think I have that right. And the only option we had from the test cross group would be little g, little d. Okay, so here we go. Let me do this really quickly. And okay, so which one would have both dominant phenotypes? Those would look like our parental group. So that would be this one right here, right? And this one would look like parental group, little g, yes. Okay, now anything in between, this would be dominant of our green, and then it would be tall, and this would be purple and dwarf. So these are what's called the recombinants, okay? All right, so at the test cross, we had 25% of each. Oops. So that was a 1 to 1 to 1 to 1 ratio. So now the question's asking us, describe how the proportions of the phenotypes of the resulting offspring would most likely differ from those of the test cross between an F1 individual and a GGDD individual recessive. Okay, so this is what the test cross results would be. So how would genetically linked, what would that look like? So I'll give you just a moment to think about it before I tell you, and then we look at the scoring guideline. And I'll give me a moment to get my clock together. Okay. All right. So when you have linked genes, you're going to have your recombinants are going to be less than 50%. So if these were 25 and 25, they're both going to have to be reduced. Okay. So they're going to have to be less than 25% each because we've got to get both of those recombinations together less than 50%. And then that would make our parental groups have to be greater than 25% each because they have to be greater than 50% each together. Okay, so let's look at the scoring guideline. All right, so the majority greater than 50, the majority greater than 50% would have the parental plant phenotypes. Okay, we got that. Greater than 25% would be green dwarf, and greater than 25% would be purple tall. Is that right? Okay, green dwarf and purple tall. Yes. Okay, we got that. And then less than 25% would be green tall, and less than 25% would be purple dwarf. Okay, and there's our green tall, and there's our purple dwarf. Okay. All right. I hope you got this right. But remember, less than 50% on your recombinants. And we'll talk about it at the very end too. I'll give you some hints on what to look for. All right, so what you need to know. All right, naturally occurring diversity among and between components within biological systems affects interactions with the environment. The same genotype can result in multiple phenotypes under different environmental conditions. Environmental factors influence gene expression and can lead to phenotypic plasticity. Phenotypic plasticity occurs when individuals with the same genotype exhibit different phenotypes in different environments. So plasticity just means a change in gene expression as a result of an environmental experience. So I'm going to take you on a little journey through environmental experiences first. This first one is a seasonal, and it's the white Arctic fox. And the white Arctic fox has a seasonal color coat. Melanocyte stimulating hormone receptors are receptors for melanocyte stimulating hormone. And melanocyte stimulating hormone controls the level of tyrosinase within melanocytes. Tyrosinase is required to make melanin. So high levels of tyrosinase are needed to make eumelanin, and low levels are needed to make light-colored pheomelanin. So in the winter, the melanin-producing pathway is interrupted. Melatonin is released during dark hours. So it's a type of photoperiodism that's going on and inhibits the tyrosine, which is responsible for the production of the melanin. I went down a rabbit hole on this little white Arctic fox, and they actually are they're heavier than this than the winter to help keep warm. All right, so assessments are always just not assessments. Assessments are, you can learn so much from assessment because not only do you have the stimulus to learn from, you have each of the choices to learn something from. And so I put a lot of questions in here that just give us a variety of things to talk about that we wouldn't have talked about probably in class. So this one is actually what we just talked about. Arctic foxes typically have a white coat in the winter. In summer, when there is no snow on the ground, the foxes typically have a darker coat. Which of the following is most likely responsible for the seasonal change in coat color? This was one of the first questions, I think, after the revision in 2012, and I've loved this question ever since. All right, so our choices are: the decrease in the amount of daylight in winter causes a change in gene expression, which results in the foxes growing a lighter appearing coat. That sounds pretty much what we just talked about. The diet of the foxes in the summer lacks a particular nutrient, which causes the foxes to lose their white color and grow a darker colored coat. The stimulus did not tell us anything about diet, so let's eliminate that. Competition for mates in the spring causes each fox to increase its camouflage. There was nothing about competition. Now, this is the one, if my kids are going to miss it, they're going to choose this. The lower temperatures in winter denature the pigment molecules. Okay, it's lower temperatures, and this is the pigment. This isn't an enzyme with the active site. It was a trick, and a lot of kids, when they see this answer, they choose that. But it's actually about the photoperiodism because we had the snow on the ground, so look, the longer nights. So what's our answer was A, yes. All right, our next environmental experience, flower color based on soil pH. My favorite flower is a hydrangea, and in a soil pH of 5.5, you'll have blue, and a soil pH of 6.5, you'll have pink. But if you have a mix, then you have purple. Here's another environmental experience example. Low population density, probably. Sex determination in reptiles, uniparents, asexually reproduce. They reproduce through parthenogenesis, which means the egg develops without being fertilized. It could be a rare response to a low population density. All the lizards are females. There are no males. Ovulation is stimulated by courtship mating behaviors, and they alternate 2 to 3 times per season per male and female roles. When estradiol is high, they act as female. When progesterone is high, they act as males. All right, another, well, I think it's our first temperature one. Environmental experience temperature, we have African violets in the form of a question. All right, so an African violet grower observes that genetically identical African violet plants grow near the walls of the greenhouse that have white flowers. I'm going to go ahead and do this. This is our greenhouse. Okay, the plants, okay, what did it say? The walls had white flowers. I'm going to go ahead and do that. All right, the plants growing farther away from the walls have pale blue. That's not very pale, but it's paler than where I'm going. Okay, and the plants growing nearest the center of the greenhouse have dark blue. Okay. All right, which of the following best explains the differences in flower color of the African violets in the greenhouse? So, warmer temperatures result in genotypic alterations, which result in flower color differences. I'm not going to choose this one because this temperature doesn't make the genotype change right there on the spot. The plants along the walls of the greenhouse are homozygous recessive and therefore have white flowers. Well, if we had a recessive, then we have a dominant, and we have more than one, so I'm not going to, and there was no mention of planting them in any particular way. An enzyme responsible for flower color does not fold correctly in cooler temperatures, and the greenhouse is warmest in the center. Okay, let's hold that thought. And more light is available along the walls of a greenhouse, so the flowers need less pigment to absorb sunlight for photosynthesis. Now, if it's greenhouse, the whole thing is green. Every wall is green and the roof is green, or if it's clear, it's all going to be the same. So, and less pigment for photosynthesis, none of that makes any sense. So this one right here, and I actually went on a little look for enzymes not folding correctly in colder temperatures, and I did find some research that included those statements this morning. All right, environmental experience, example temperature again, sex determination in reptiles. Australian dragon lizards have a ZW sex determination system. The male genotype is homogametic, ZZ. So here we have our male is ZZ. And a female genotype is heterogametic, which is female, and she is a ZW. However, all eggs incubated at temperatures above 32 are female, okay? Which of the following best explains how the development of the phenotypic female Australian dragon lizards with a ZZ genotype occurs when incubation temperatures are at above 32? All right, lizard embryos with a ZZ genotype cannot develop. Well, we just said that they could, okay, because they're going to become female. So it's not it. An incubation temperature above 32, Z's do not turn into W's, okay? Just because the temperature, a gene doesn't change on a chromosome because of that. At incubation temperatures above 32, crossing over transfers genes from the W to the Z, producing females. I think that we already have, all eggs are incubated. It's already an egg. That's already happened. And incubation temperatures above 32 inhibit the genes on the Z, okay, on the Z, and that produce proteins necessary for male. And so that is our choice. Okay. All right, and here's another one. I have several of these, but it's fun to talk about these things that we wouldn't usually talk about. Environmental experience, example, light, color, sex determination in reptiles. So a scientist studying phenotypic variation in a species of butterfly observed that genetically identical caterpillars grown in similar cages but exposed to different colored lights developed into butterflies with differences in wing color and body size. So here we have a white wing color darker when it's red, okay, and I should have grabbed a red. There's red, and we have smaller when we're red, and then with blue we have a lighter wing color and we have a larger size. Which of the following best explains the cause of the phenotypic variation observed in butterflies? A, different mutations occurred in the caterpillars that were exposed to different colors of light. No. If they had mutations, there's no guarantee that all are going to have the same mutation, and then that would be before them. Okay, that would be in a previous generation. The energy used to grow a larger body results in butterflies with lighter colored wings. No. Individual caterpillars evolved adaptations. Now this sounds too Lamarckian. There were different gene expression of wing color and body size in response to the colors of light the caterpillars were exposed to. There we go with gene expression, okay? And yes, we have D. All right, effect of increased UV on melanin production in mammals. Humans produce melanin and melanocytes. There are two different types of melanin, and I spoke about these earlier. Eumelanin and pheomelanin. Eumelanin is very dark and pheomelanin is fair or red. And melanin protects the DNA in the skin from UV rays, which would cause mutations. The melanin forms clusters over the nucleus to protect the DNA. So what I talked about this when I used to teach anatomy and physiology. So let's do this. Here's our nucleus, and here's our DNA, okay? Here's our DNA. And the melanin pigment, so here's the sun, the melanin pigment will cluster to protect the DNA in the nucleus, in those cells. Okay, so UV rays were mapped, and higher UV rays are in areas of the world where darker melanin can be found. There is a correlation between the intensity of UV radiation and skin color. Where UV is strong, skin is dark. Pheomelanin is found where UV is not strong because sunlight needs to be absorbed to produce vitamin D because the sun is farther away from those areas. So less pigment is necessary to get more sunlight because there is less UV. So I absolutely think that you should watch the biology of skin color from HHMI. It's just an amazing little short video. Make sure you do that sometime. All right, so I know I drew this before and on, I think it was signal transduction pathways. So let's do it again. And we have, let's see, what did we have? Let's do it over here. Here is our A. So example, presence of opposite mating type on pheromone production is what we're doing now. So that's going to have, that's going to turn on our gene, okay, if we're a yeast. All right, so here we are, and we are, let's do this. This is the alpha cell membrane. It's not bigger, just gives me room to work. And here's a receptor. And this A yeast, A-type yeast, is going to release its mating pheromone, okay? And it's going to bind to that receptor. And then, I'm going to make this a little bit bigger. And then we're going to have this event, and I'm going to phosphorylate some of these in a moment. Okay, so you know how this went. Let me get this to a 5. Okay, All right, so we have this signal, this was reception, signal transduction is going to happen, phosphorylation is going to happen, and it just continues. And then we have these phosphorylated molecules at the end. Well, it's going to, here is a actin filament, which is responsible for the structure of the holding the shape of the cell. And this particular molecule is going to put actin molecules on the actin filament. So when this is reception and then the signal, and then this is going to go to work and start making these actin molecules, genes will go to work, proteins will be made, actin molecules will be made, and there we have our shape that we're going to call a shmoo, all right? So it will then reach this A, this alpha wants to reach this A because it's of this mating pheromone, okay? All right, now, off of the environment, so this was environment. This was an environmental experience because the mating pheromone was in the area. So that's the end of the environmental experiences. All right, next we're going into chromosomal inheritance, non-disjunction, and then some other patterns that are a little different. Okay, so these are going to be reviewed, just review. These pictures are bigger. If you look in the Mendelian genetics slide deck, you'll find them there. Anyway, so look, chromosomal inheritance generates genetic variation in sexual reproduction, segregation, independent assortment of chromosomes, and fertilization result in genetic variation in populations. The chromosomal basis of inheritance provides an understanding of the pattern of transmission of genes from the parent to the offspring. So if you remember, we talked about law of segregation, and we had a big D here and a little d here, and this would never be, this little d would never be expressed as long as it's with that big D. But during anaphase of meiosis 1, we have segregation. We have the separation of the big D from the little d, and then this is often a cell by itself, and this is in a cell by itself. And then if we have a cross with another of the same little d, then we're going to be able to be expressed as in that 3-to-1 ratio of our monohybrid. Okay, as far as the law of independent assortment, when we had our meiosis 1 meiosis 1, metaphase 1, and anaphase 1, then we were going to split down the middle, right? That was a bad down the middle. And anyway, so this originally had big D and big R. Do they have to go that way and little d with little r? No, they do not. All right, so we can have big D with little r. So this could have been over here, that could have been over there, and vice versa. There is no order that this has to go in. It's random, long as their homologous pairs are next to each other. And that's the law of independent assortment. And we can see that in our dihybrid cross of 9331. And then we have our meiosis, we have fertilization, and there's a whole slide deck on meiosis. And if you have, here's 3 pairs of chromosomes, they go through interphase and they replicate, then they go through crossing over. Remember, that was a step of genetic variation happening right there. Independent assortment, we had variation going on. Anaphase, now we have 2 cells, and then we have anaphase again. And now here, these 4 daughter cells are so different than this original 3 pair. And when they cross with another diverse offspring, someone else's other offspring, then we start this all over again. And genetic diversity just increases generation after generation. Okay, certain human genetic disorders can be attributed to the inheritance of a single affected or mutated allele or specific chromosomal changes such as non-disjunction. So first we'll talk about single mutations, okay? So if this is messenger RNA, I can say that it's going to look exactly, other than the U, it's going to look like our coding strand of DNA. So I'm just going to pretend that this is our coding strand. It would be GAG, and this would be GTG. Okay, if we had a coding strand with GAG, then we're going to code for glutamic acid. and have normal hemoglobin. If we code for GTG, then we're going to get valine, and we're going to have a mutated hemoglobin. So now let's take this, the next slide will actually take the messenger RNA to this. This was the mutation right there. Should be an A, but was a T. So let's take these messenger RNA to the genetic code. All right, so we have GAG, so we're working with normal, and that would be our GAG is right here. There's glutamic acid. So what's going to happen is this GAG will have an anticodon for CUC and bring in glutamic acid. Glutamic acid will then be added to this polypeptide chain, and that's what's supposed to happen. It's the normal hemoglobin. On the other hand, if we have a GUG, then it's going to code for CAC. Here's GUG, and there's valine, and it's going to code for valine, and valine is going to be added to that polypeptide chain, and that is the problem, okay? It's not supposed to be valine, it's supposed to be glutamic acid. And if you can see, glutamic acid is hydrophilic and valine is hydrophobic. If we have a protein and you are hydrophilic, that means that you can be found on the outside of the protein, because these are amino acid, these little dots, because you are polar. And then hydrophobic would want to be on the interior away from that water. So that makes a big difference whether you're on the outside or the inside. That means this is going to fold improperly, and the result, the function is not going to be the same. So what happens is you have this sticking together of these molecules that make this sickled shape right here. All right, Huntington's disease is another single affected allele. A genetic counselor is consulted by a young man who is worried about developing Huntington's disease, an inherited disorder caused by a dominant allele of a single gene. The young man explains that his cousin was recently diagnosed with Huntington's disease. Let's go ahead. Inherited, and we're talking about a cousin. And the news has caused him to consider his own risk of developing the disorder. Which of the following questions will help the genetic counselor to evaluate the risk of the young man developing Huntington's disease and transmitting it to his children? So this is a question about questions, is what I call it in my classroom. Okay, were you and your cousin born in the same geographical area? Okay, this was an inherited disorder. It wasn't due to an environmental experience. Okay, were your parents or grandparents ever diagnosed with Huntington's disease? This would be a good question. Were you in physical contact? No, it's not contagious. And were you ever exposed to substances that are suspected of being mutagens? No, this is not about getting this as a cancer, okay? So Huntington's disease, and let's see. And B, it was. So I put this slide in because I wanted to mention some things about this. So it said it was autosomal dominant, and it's lethal, though it's later in life after 40, and it's a degenerative disease of the nervous system. Okay, so now that was our single allele, mutated allele, and now we're going to talk about non-disjunction. So this is in anaphase of meiosis 1. And so what should happen is that these homologous chromosomes would be pulled apart, and these two would go to that pole, those two would go to that pole, but instead they stay together, okay? They're still joined. And what happens is there's only 2 chromosomes. There's one on this side and one on this side when we get to anaphase 2. And then there's 3. Then we have one of each of these, 1, 2, 3. So at the end in our 4 cells, we have N+1, which is 1 too many, N+1, 1 too many, and N-1, 1 too less, N-1, 2 less. So all of these have a chromosomal abnormality, okay? Now, in the second type of non-disjunction, we're going to have this happen in anaphase 2. And if you'll notice this right here, so let's stay, so far so good. So we have our 2 homologous pairs, they separate, that's good. And now these two sister chromatids need to separate, but yet this one didn't, okay? It did not separate. So we have normal, normal, everything's normal here. So we have at the end a normal cell with those two, normal cell with those two. We're missing one, so it's n-1, and we have one too many, and we have n+1. Okay, an example of non-disjunction is trisomy 21, which is Down syndrome. It's an aneuploid condition, and it has characteristic facial features, short stature, correctable heart defects, developmental delays, risk increases with mother's age, and most are from meiosis 1. And this is what it looks like. So this is a karyotype. So on a karyotype, you have all of your autosomes for a human being, 1 through 22, all pairs. There's 2 of each, and they're organized according to their length. And there's some bands and things like that that come into play. But this one right here has 3 chromosomes instead of just 2. So this is why it's called trisomy 21, and this is Down syndrome. And here we have, that's a girl, that's an X would be a girl, but a Y makes this a boy, just an FYI. So this is a boy with Down syndrome. All right, non-disjunction Klinefelter syndrome. Let's look at this karyotype before we read the question and see if we can tell what's wrong. So we have everything is looking normal. We actually do this karyotype activity in my classroom with this magnetic board, and it's great. We have different scenarios in little bowls, and they switch around between groups of which chromosomal problem they're doing. Everything looks good until we get right here, right? So we have 2 X's, but we have a Y, so that makes us a boy, okay? The diagram above depicts a karyotype of an individual human. Which of the following statements concerning the karyotype in the diagram is true? A, the diagram illustrates a condition found in females. Is that right? Nope. The diagram indicates a mechanism for increasing genetic diversity. This is not a good mechanism for increasing genetic diversity, okay? The diagram illustrates results of non-disjunction during gamete formation. Yes, that's why it's there. And it indicates Down syndrome. No, we know that Down syndrome would have to do with 21. So C would be our answer. Okay, and I also wanted to tell you about another chromosome non-disjunction problem, and it has to do with Turner syndrome, and it's a girl, but she only has 1 X, and so there's nothing here, okay? And that's also from non-disjunction. Okay, so this was a lesson to myself to practice what I preach today. So look at this diagram and then read. So what we have here, we have an X and a Y, and so they separate in meiosis 1, that's good. And then they're into 4 daughter cells at the end with a haploid, okay. So figure 1 illustrates the X and Y chromosomes during meiosis 1 and meiosis 2 of normal spermatogenesis. That's what I didn't read. I didn't read this first part. I just jumped in here, and then I spent 5 minutes trying to figure out what was wrong with this picture. Anyway, if the normal spermatogenesis is disrupted, the gametes can have different chromosomes than expected. Which of the following is the most likely cause of one of the 4 gametes having 2 X chromosomes and one having neither an X nor a Y? So let's do this. Okay, so there's our 1X. Let's add this 1X to it. And then let's take out this Y. I mean, this other X. I think that's what it asks us to do. And then answer the question. So which of the following is most likely cause of one of the 4 gametes having 2 X's? There's 2. Okay, so they've just been through meiosis 2, shouldn't have that. And one having neither an X or nor a Y. Okay, so we have empty. Non-disjunction of the chromosomes during meiosis 1. Let's see, something's wrong here. Oh, I'm trying to write in white. Okay. All right, did anything happen wrong here? No, we're still good. That's what we should look like. So that's not it. Non-disjunction of both the X and the Y, not affected Ys, not it. Non-disjunction of the Y, not it. Non-disjunction of the X chromosome during meiosis 2. This one still looks like this one, okay? So that is the answer, okay? And don't be like me and not read before I answer the question. I kind of learned a little lesson today. Oops, let's go back. Okay, last one of these scenarios, okay? Red-green color blindness in humans is caused by a recessive allele located on the X chromosome. Figure 1 shows the potential offspring of a female who is red-green colorblind and a male with full color vision. All the possible male offspring would be colorblind, and all the possible female would have full color vision. If during the production of male gametes, an error, so we're to this paragraph right here, was referring to this. If during the production of male gametes, an error in meiosis occurred, sperm containing both an X and a Y chromosome could be produced. So it took me a minute to figure out what this was, but this is just a Punnett square that they have here, okay? All right, so possible offspring. Now, what does the question ask us? How would the extra chromosome, so we need to look for where's there an extra chromosome? It's right here, okay? And let's look at that. If we have a Y, what do we already know we are? A boy and a boy, and we have a big B, so we know we have normal vision. So that is what our trait is going to be, 2 traits. So how would the extra chromosome affect the male offspring produced by the gamete? None of the potential offspring would be male. I think that's wrong. The male offspring would all be red-green colorblind. No, we already figured it out that they're going to be normal. The male offspring would have full color vision because of the presence of the extra X. Absolutely, there it is. There would be no change. Yes, there is, okay, of the possible offspring because the extra X chromosome would not be active. That's not true. So our answer here would then be C. Okay. All right, so now on to a new topic, chromosomal changes, and this is deletion. We have an A, B, C, D, E, F, and G, and if we were to take out this C, if it was a gene or a segment with several genes on it, then we're going to lose the gene expression that's happening right here, okay? Deletion is a problem. The next one would be a duplication. If you see we have A, B, C, D, E, here's our C, but now we have a segment repeated. And we'll talk a little bit about that at the end. And then an inversion. So this ED, the CDE, just does a total flip, and now it's EDC. So it's reversed, and there's going to be an imbalance in its gene expression. And then we have a translocation. Here's our translocation. We have our A, B, C, D, E, F, G, our H, I, J, K, L, M. Now look after the homologous chromosomes have separated. Here is our H, I, J, and here is our K, L, M on 2 totally different chromosomes, okay? And this will also cause some differences in gene regulation. Okay, some guided practice. We're gonna write just a very short FRQ today. All right, science skill practice, predict the causes or effects of change in or disruption to. We've been doing that through several multiple choice questions. One or more components in a biological system based on a visual representation of a biological concept, process, or model. And the task verbs that we're going to do today are identify, make a claim, provide reasoning, calculate. I think I took out a lot of these, especially calculate. I built this presentation way too big, but I did leave some great questions in the slide deck if you use it, okay? And that's under natural selection 7.1, our very first presentation. There's a link to the slide decks. Okay, so writing FRQ responses. Meiosis occurs in many organisms as part of sexual reproduction. So we're going to discuss, and 2 of them, examples of mitotic errors and their effects on offspring. I'm going to give you 30 seconds or so, maybe a minute to think about this and jot down some answers. We're doing great on time, so go ahead and think about this. Okay. Did you jot something down? All right, so you should have said, so you should have said non-disjunction and then the chromosomal changes. So non-disjunction, spontaneous abortion can actually occur because of this. Monosomy, that means the one that we were talking about, like Turner syndrome. Trisomy, we talked about, was Down syndrome. And anything that's not your normal N number, that's more, is called polyploidy, okay? Chromosomal changes, here's the 4 that we talked about: duplication, deletion, inversion, and translocation. But this scoring guideline seems kind of like out of whack a little bit, so I did my own. It looked like the right answers are there, but just not in the right order. So a deletion would be loss of critical gene expression, and an example of that was Cri-du-Chat, and I learned about that in nursing school actually. And it is a male that has this cry of a cat. And duplication, Fragile X, and there's some other repeats that are problems when there's duplication. And inversion, there's an imbalance of the gene expression. There's a 180, you know, we had CDE, and then all of a sudden it went EDC, okay? And I read also that there were some inversions that can possibly connect with differences between us and chimpanzees. I read that this morning. All right, translocation, change in gene regulation. There's a condition called Philadelphia chromosome. I actually have my students go on a little web quest, like search about this, and it's about chromosome 9 and 22, and it causes chronic myeloid leukemia. All right, so debrief and summary. Today's takeaway: naturally occurring diversity among and between components within biological systems affects interactions with the environment. You should be able to explain that the same genotype can result in multiple phenotypes because under different environmental conditions. And some chromosomal inheritance generates some genetic variation also. So remember, this was the different environments, and this we talked about non-disjunction, we talked about mutations, and we talked about what else? And some chromosomal changes, okay? All right, predict the causes or effects of a change in or disruption to one or more components in a biological system based on a visual representation of biological concept, process, or model. And we talked about, we predicted what would happen if it was different almost the whole day through this. Stay tuned because I have some practice exam information for you at the end. So it's a common errors. Now, where I get my common errors from is my classroom, what I see happening, and then from myself, because I do everything I have my kids do. I have every struggle they have. Things are not always easy for me. And so I feel their pain sometimes. But I want to give you one thing here. When you see these, now let's just say this, for example, 433, 433, 55, 55. I don't want you to think about it. You already should have linked genes in your mind, okay? Because you can see that this is less than 50%, okay? 50% recombination. All right, then a dominant trait is not necessarily a favorable trait. Remember the green and the dwarf and the tall? I immediately, without read, because I didn't read, I had D going with tall, okay? Because it just seemed like the normal version would be the dominant trait. That wasn't necessarily so, okay? Read questions carefully. The diagram, this was the other one I spent a long time on because I didn't read that was the normal version. I kept trying to find what was wrong with it. So do as I do, and do as I say and not as I do, I guess. All right, hold on. So here we have some homework for tonight. AP exam practice homework. You've seen this before. A spermatocyte produces the following four sperm cells. These cells are a result of non-disjunction during which of the following phases. Real easy homework. I wouldn't just go jump back to where this is in the presentation. I would take and draw out meiosis yourself and follow those chromosomes and then figure out how to get to here. Like put this at the bottom and then work backwards. Okay. All right, so your to-do list. Check your device and your internet connection. Check your email address and your phone number and AP account. Tell your teacher you are testing and breathe because your test ticket will be in your account also. It'll be in your email and in your account. Next review session, we're actually going to start Unit 6, which is our last unit. And we hope you'll join us again. Stay tuned though, because I'm about to go over some optional practice FRQ information. So let's do that really quickly. We're good on time. All right, so this is for everybody to know. So you need to go to your AP classroom and log in. So that's your first step is log in. And then you're going to click AP Classroom up here in the right. And then you're going to click here for optional student practice. And look, you have access to all these videos all the time without going to troll through YouTube looking, okay? They're all a link right here. Now click on this, practice answering free response exam questions. And then look for this sign, this green button, okay? It says optional student practice. These questions marked with this symbol mean that you can take the question, you can answer these questions on your own. Your teacher does not have to assign them, that you can do this on your own for your own practice. So if you click on one, and this is biology that you're looking at, but the same method goes for all of your classes if you have other AP classes. So this doesn't tell you what the content is about, but it tells you about science practice here. Okay, so we're going to click on one. And then it's going to open up and we see some graphs. This is biology. They did biology as the example for everyone. And then we're going to type in our answer into the box, okay? And then we're going to hit this small little button here that says submit. And then, are you sure you want to submit your assessment? You're going to still say yes. And then this is where I want you to stop, okay? See how this question is scored on the AP exam. Before you submit, would you like to review how this question might be scored on an AP exam? You can review and optionally revise your response. So this is what I want you to do. Yes, review, okay? Because this is going to bring up the scoring guidelines. And you know what you wrote and you know what you should have written. And so now you can actually go back and you can have the opportunity to revise your answer, and then that gives you the experience of writing out the sentence in the proper format and include all of the content that you needed to include. So after you revise it, then you can hit the final, I'm one ahead of myself actually here, then you can hit the final yes review, okay? Well, that was the yes review. Now you're going to hit submit because you've already reviewed on the last slide because I was ahead of my time. Okay, and I think that's it. All right, so I hope that helps. Go find those questions and then come back tomorrow and we start Unit 6. Okay, thank you. Good night.